Cell cycle can be distributed into two parts, the first one being the interphase and the second one being the M phase. Interphase is the time where the cells do a lot of the preparation before the actual cell division that occur in M phase. In fact, the cell will spend 90% of its time in interphase and only 10% of the time in the actual cell divisions. You can think of this just like your exam. You will spend 90% of the time in preparation of the exam and only 10% of the time in the actual exam. Interphase begins with G1, follows by S, and ends with G2. Meanwhile, for M phase, it begins with mitosis and ends with cytokinesis. So the cell cycle will look something like this. G1, it means gap number 1 or growth 1. This is a time where the cell has a very high metabolism rate and it grows very fast. On top of that, cytoplasms, organelles, protein and enzyme are also being synthesized. Synthesized eventually just means produced but chemically. And most important in G1 is the decision-making process, where the cell need to do decision whether they want to continue with the cell division or not based on whether they have enough of resources or the environment allowed them to do so or not. If the environment is too harsh, then they wouldn't continue with the cell divisions. How I remember what happened in G1 is by OMG. M stands for very high metabolism rate, G stands for they grows very fast. And I saying that if you say OMG, you need to cope the stress. So coping the stress by COPE, which is our cytoplasms, organelles, protein and enzyme being synthesized. Once G1 is done, it will go into S phase. S stands for synthesis. This is where the replication of DNA occurs. In interphase, chromosome eventually exists as chromatin, where it's just a thin and long structures. This is where it's in the form of uncoil. This chromatin will eventually duplicate itself, which is copy and paste, so that we have one more identical copy that can be passed down into our daughter cell later on. So basically, replication of DNA is just a process of copy and paste of the DNA informations. Once the copy and paste is done, it will go into G2, which is represented by gap 2 or growth 2. So basically, whatever happened in G1, it will repeat in G2, except about the decision-making process. Instead of decision-making, they will now try to accumulate as much energy as possible for the final preparations. You can think of G2 is the night before your exam. You try to store as many energy as possible and do the final preparations so that tomorrow you won't be sleepy or have the problem with the exam. So this sums up the interphase. Now, before we look into M phase in the next video, we're going to learn some of the new terms, which are chromatin, sister chromatid, and chromosome. As we know, chromosome can be found in nucleus, but chromosome appears in different form in different stages. For example, during interphase, chromosomes exist as chromatin. Chromatin is whenever the structures of the chromosome is thin and long. It's same as the name, thin. So you will see something like a straight form, like a hair of the chromosomes. This is a time where the chromosome is uncondensed, means they are not condensed, and they are uncoiled, means they are not curled up. So it will look something like this, where it's surrounded by the nuclear membrane, or sometimes refer it as the nuclear envelope. And each of the chromatin has the centromere. This is the part where later on two chromatin can connect to each other. During S phase, this chromatin will undergo the replication of DNA, where we will copy and paste the identical chromatin which contain the same genetic informations. So once copy paste is done, it will bind together at the centromeres. At this stage, we are no longer refer this as chromatin, but instead we are now calling it as sister chromatid. So we have one sister chromatid on the left and another sister chromatin on the right that bind together in the center of the centromeres. So as we can see, two sister chromatid will make only one chromosome. Now, let us go deeper about replication of DNA during the S phase. As we know, human consists of 46 chromosomes, but during interphase, they are eventually exist as the thin and long structures that we refer as chromatin. So we have 46 chromatin with the 46 centromeres. But in S phase, replication of DNA occurs, where we tend to copy and paste the genetic information of the chromatin, where it looks something like this. So now, instead of referring it as a chromatin, we're now saying that we have two sister chromatid that bind together at the centromere to form a chromosomes. So we are now counting it by sister chromatid. So don't forget, two sister chromatid make one chromosome. So after replication, we have 92 sister chromatid. But then, we still maintain the same number of chromosome, which is 46 chromosome. Hey, if you want to join my online tuition class, please drop me a message on Telegrams. Or if you want to support us so that we can make more videos like this, the simplest way is just to sharing the video with your friends. Hit the like buttons and consider subscribe to this channel. See you in the next video.